Right, so I'm here on behalf of Holger and me. As Aviv has mentioned, Holger had all the intention to be here even yesterday, but his first flight was delayed, and then he's stuck somewhere in the Paris airport. And hopefully he's on a flight now so that he can join us later today. So too bad he couldn't be here because he prepared most of the slides. So I'll see. I just contributed a few. I'll see whether I can do as good of a job as he would have. So it's about the.
I'm just going to turn on your microphone.
Oh, no wonder. I was wondering. Okay. All right. So the only most important thing that I said is if Holger had Holger been here, he'll give a better talk. Okay. So this is about the Standard and Technology Working Group, and this is the composition of the Standard and Technology Working Group. We have 17 members from 14 institutions, 7 countries, and 3 continents. Look at all the statistics that Holger did. And covering all these various different approaches that are really important for generating a more complete human cell atlas, or for that matter, cell atlas of many different kinds of model organisms too. So a few important things that our working group is working in trying to I was asking Holger, why do we have a cell atlas logo like that, look a little bit fainted, and then like that? He said, our working group will make the cell atlas shine. So I was on the phone with him yesterday. Now I understood this contrast by doing the following, and that is, we'll make sure that the data are reproducible, so that if the cell atlas as a whole initiative, we report an initial cell atlas, other people can reproduce it and find exactly the same results and profiles. Of course, some improvements we always expect as time goes on. And high integrity means that we can capture appropriate cell proportions, and also the rare cell types. And then also predictive value that we can map these molecular profiles either of a known cell type or actually predict new cell types. And this is the mission of our working group, and I'm just going to read it. We want to define standards for single-cell data production through initiation, guidance, and analysis of benchmarking projects. I'll highlight more about the benchmarking projects. And we will support benchmarking and scaling, supporting benchmarking and scaling of emerging single-cell technologies that are relevant to key HCA areas. And of course, an important thing is we'll do our best to disseminate the standard operating procedures and the best practice for high-quality single-cell data production. So here are some of the action items that we have. This one I will highlight more. We'll be benchmarking various of different kinds of projects, compare to see the pros and cons, and see how to combine them to better get an overall complete cell atlas. And then we'll be working closely with the analysis working group. And then together we come up with the quality control metrics in an iterative manner. And then we will also be coming up with the standard operating procedures for sample preparation, because as you all know that your sample prepared in different ways, in different hands, you often come up with different kinds of results. And then there are many different kinds of approaches, and we will be trying our best to find a coherent way in terms of integrating these platforms. They really can happen at various stages. They can happen at the very beginning, even before we start taking data, because a priori or based on experience, we would know that certain methods, they can feed information into others and vice versa. So then in the experimental get-go, we want to actually have designed it in a way that actually it is a coherent approach that combining different approaches to take a more complete data. But also, when the data are generated, we want to have this integration to combine different data sets together, especially with the help of the analysis working group to put everything together into a more complete picture. And then eventually we want to sort of, especially for some of the new emergent methods where the standard data modality isn't exactly clear yet. For example, I work on MERFISH, and I wonder, what type of MERFISH data information is needed in that thing that we want to actually put onto the DCP. So this is some of the missions and action items of our working group. And then I thought I'd highlight two of them, benchmarking and platform integration, okay? So in terms of benchmarking, there are already several, sort of program projects or consortiums or collaborations that we can think of, but there are probably more. One is the Space TX Consortium, and the PI is at Lane at Allen Institute, and this is supported by CZI. And then there, they put a lot of different spatial transcriptomics approaches together, analyzing the same data, imaging the same data, analyzing the same data, and then so that we can actually compare the strengths and differences of various spatial approaches. And then, of course, in a goal of accelerating the development of this. And then there is a proteomics and the transcriptomics comparison led by Emma Lumberg. And then that is comparing and benchmarking proteomic and transcriptomic-ish analysis of the same tissue. And then there are two collaborations or consortiums that are comparing single-cell RNA-seq methods. And one compares 7 of them, and then using fresh sample, and then the data is processed by a single lab. And that's led by effort by Josh Levin at Broad. And another one is a comparison of 15 single-cell RNA-seq technologies. I really have to keep up with all these single-cell technologies. Using cryopreserved sample that Holger said he had already distributed to different labs, and then they're going to be processed actually in individual labs and so on for comparison. And then the idea is with this, we can come up with not necessarily the best, but seeing what's the pros and cons, and then how to combine different approaches to put together a better picture. And then, of course, one could imagine many more such collaborations or consortiums or program projects that define the quality metric, quality standard, and standard protocols. And even not only evaluating, maybe there will be new technology emerging from these consortiums. Now, another item on this diagram that I want to emphasize is the platform integration, because it is really, we more and more see that there are complementarity between different approaches. And then if you actually coherently marry them, then you really can learn more. And then I selfishly use our own work that is fresh out of the oven, and then it'll be in press at 2 p.m. today. And Laura told me that it's okay to show a little bit of a snippet of it ahead of time. So basically, it is a collaboration between Catherine Dulac's lab, Aviv's lab, and my lab, where we combine 2 approaches, the single-cell RNA sequencing and MERFISH, to create a cell atlas of an important brain region. That's the preoptic region of a hypothalamus, which is really controlling a variety of important social behaviors, some listed here, parenting, mating, and aggression, but also a lot of homeostatic functions, thirst, hunger, and sleep, and so on. And the idea is, as many of you know, that single-cell RNA-seq is an unbiased gene expression profiling approach. So it not only can give us a catalog list of the cell types, but also it can help defining molecular signatures. And then with that help, then some of our Murfish then is an imaging-based approach where we do single-cell transcriptome imaging, but then that's targeted because we select a set of genes to image individual cells to profile them. And then, so the gene selection, we get help from the single-cell RNA-seq. Half of the genes are selected based on cell markers by the single-cell RNA-seq, or defined by the single-cell RNA-seq. The other half is based on known knowledge of the hypothalamus in terms of what are the genes that are of functional importance. And then we did the Murfish, and then combine these two, that we actually have some, not only the major cell classes that we get them all, but also even in neurons, for example, we get 70 different kinds of neuronal types. And then there, the combination really makes sense because we would have, the majority of them actually have good correspondence between the two, but we have some cell types that we get from Murfish, but not single-cell RNA-seq, perhaps because they are defined by the low expression receptors and so on that are more difficult to define or measure quantitatively by single-cell RNA-seq. And vice versa, we have clusters that we can see with single-cell RNA-seq, but not with Murfish, presumably because their marker genes were not in our 155 gene list for Murfish measurement. So with that, we get a more complete map, and then we also, because Murfish is an in situ imaging method, we directly get the spatial organization. And also because Murfish is a in situ imaging method of intact tissue, we can combine that with activity marker imaging to directly tell us which cell type is responsible for which behavior by including, for example, immediate early gene that gets activated by parenting. Then we say, these are the parenting cells have this kind of a gene expression profile. So then together we get what we considered a molecular and spatial and functional cell atlas of the hypothalamus preoptic region. And a beautiful, we have many, well, of course, it's at large volume, actually, three-dimensional volume. This is just one slice, but even looking at this one slice, I could look at it a long time and appreciate the beauty of this spatial organization of cells in our brain. And then I imagine that the spatial organization of cells in any of our organ will be all that beautiful. So this whole human cell atlas will just give us enforce us in understanding how beautiful human and animals and plants and so on are. So with that, then this is the final slide to tell you how to communicate with us. And then there's a Slack there. You can download data. You can upload your analysis results. You can participate in discussions. And then we welcome you to put your protocol into protocol IO. And then also, if you have any questions and comments you want to contact us, this is our email address. And thank you for your attention.